In this experiment, you're going to be given a few things. You're going to be given a 12 well strip, as you can see right here. You're also going to be given wash buffer. And then in addition, you're going to be given a lot of test tubes. So the yellow ones contain your bodily fluid sample. The pink ones contain the positive control. The purple contain the negative control. The green has the primary antibody. The orange has the secondary antibody, and the brown has the TMB substrate. For this ELISA that we're doing, the antigen is the BASP1 protein, and then the primary antibody would be the platypus anti-BASP1, and then the secondary antibody would be goat anti-platypus with conjugated HRP. Throughout the experiment, the things that are going on are going to be represented by these kind of figures. So if you can imagine, this is a side view of the well, so we can kind of see what's actually going on. And the first thing to really point out is what this well actually does and what it's made of. So if you look down closely at the molecular level, it's coated in a polymer called polystyrene. And as you can see, it just has a bunch of aromatic rings sticking off of it. So it's really hydrophobic polymer. And what this is going to do is it's going to allow for hydrophobic or van der Waals interactions with proteins. So the outside of the protein is hydrophobic. So essentially what's going to happen is the protein itself is actually going to just stick onto the walls of the well. And this is what's going to actually help us to keep the protein in the well for the duration of the ELISA assay. Aliquot 50 microliters of the appropriate sample into the correct well. In this stage, we are getting the antigen, if there is any, to stick to the well. The next step is to remove the sample from the wells. So what you're going to do is you're going to get some paper towels and then just flip over the well strip and essentially just get out all the liquid that's in there. To use a paper towel just to help to wick everything away. The next step we're going to do is actually wash out the wells with the wash buffer. I think it's important though to point out what is actually going on here. So this wash buffer has a detergent called Tween 20. You can see its structure right here. The more important part is that you can see that it has a long hydrophobic tail on it. Now why this is important is because this hydrophobic tail can interact with the polystyrene that is coating the side of the wells. Right now there is antigen that could be stuck on the wells, but there's still a lot of open space that more proteins can stick to. And in the next steps, we're going to be adding primary and secondary antibodies, which are proteins in themselves. And we don't want those sticking to the well, we want them only interacting and binding with the antigen. So what we need to do is we need to block off any remaining interaction sites that are on the surface of the well. And so adding the tween here does this. It accomplishes it, so the tween will essentially block off any remaining hydrophobic interaction sites that are available on the surface of the well, so that we don't get any non-specific interactions later on. So to wash out the wells, you're just going to add 200 microliters of wash buffer to each, and then the same way that you did with the antigen, you're just going to flip it over onto a paper towel, and just to get out all the liquid and wick out everything. You're going to need a lot of paper towels for this experiment, so just get a bunch at the beginning for all this. And we're doing 2 wash steps here. So just do it again, add 200 microliters, and flip them over on the paper towels. Next, we will add 50 microliters of primary antibody to every single well. So just like before, we're going to get some paper towels and remove the primary antibody by just hitting on the paper towel and wicking everything out. And also just like before, we are going to wash the wells with 200 microliters of wash buffer twice. Again, in between removing the wash buffer on paper towels. Next, we will add 50 microliters of the secondary antibody that has the conjugated horseradish peroxidase to each sample. And just like before, we're going to remove the secondary antibody out of the wells on the paper towel. So now you're going to need to wash the wells. Here I'm using something called a repeater pipette. It's a really cool little device where you just drop a large amount of liquid and you can set it to dispense a certain amount. I'm using it here because I was getting really tired of pipetting a bunch of times with this, so I'm just using it for the washes here. But remember now, for this wash step, you are going to actually wash it 3 times. We really want to make sure that there's no leftover secondary antibody around, because the secondary antibody is what's going to react with the substrate. So we really only want anything that's just interacting or binding with the primary antibody, which should be binding with the antigen. Whenever you get to adding your third and final wash, wait at this point until everyone in the class is at this point. The next step is time sensitive, and we need everyone to start the last step at the exact same time. And so the last step is adding the 50 microliters of TMB substrate to each sample. Like I said, this is time sensitive. You can see how quickly the colors are changing. So we need to be able to actually measure everyone's at the same time. So this is why you really need to stop at the third wash step. Once everyone is there, you can then empty out that third wash, and then everyone can add the TMB substrate, and we can go and measure it on the plate reader.